Hi everyone, welcome to my channel. For this video, I will talk about this paper, which describes how the role of antigen processing is involved in the fight against hepatitis C virus. This paper was published in November 2012 by some of the big names such as Robert Tim and Paul Kleineman, and I've also heard about Julia Schmidt as well. It's an interesting paper. It's also a widely cited paper in the field of immunity against Hep C, and so it describes the role of proteasome and antigen processing in fighting against the Hep C virus. So hopefully you enjoy this video and enjoy this paper. So if you have not already done so, please press like to my video and subscribe to my channel. And let's dive in.
So here is a title of the paper. So if you're interested, you can just type in the title of the paper on Google and then just click on your first result, and then you would be able to download the paper in the PDF format. So let's dive in. So I'll give you a brief introduction of the study. So it has been shown that the HLA-B27 allele is associated with the hepatitis C virus clearance, and one of the reasons is because the hepatitis C virus found it difficult to escape the recognition by the HLA-B27 as well as the CD8 T cell recognition. And here is the figure which kind of summarizes how the virus is being recognized by the immune system. So here you have the antigen presenting cells, which express the gene encoded by the HLA-B27 allele, and then you have which is presenting the HCV peptide, and it is being recognized by the HLA-B27 restricted CD8 T cells, and this recognition often leads to the clearance of the hepatitis C virus, although there are exceptions. However, the virus can try to escape from this. So now the virus can introduce the mutation in this region of the genome, and this kind of escape the recognition by the CD8 T cells, or it can escape the presentation by the HLA-B27. So they can escape, but at the same time, it kind of compromises the virus fitness. So now we know how the virus, can mutate, and at the same time, come at a cost of its survival. But what we don't know is the role of the immune system in all this. And this study is trying to address this. So that aim was to elucidate the immunological factors that mediated the protection afforded by the HLA-B27 allele in HCV infection. And there are three main methods that they used to investigate this. Was they stimulated the peripheral blood mononuclear cells, which are white blood cells, minus granulocytes, with antigen and peptides. They also use flow cytometry to analyze the cell population, mainly the CD8 T cells, as well as the in vitro proteasomal peptide digest and mass spectrometry. So in their first results section, they wanted to investigate the functional avidity of CD8 T cells that were restricted to either the HLA-A02 or HLA-B27. So they used these 3 HLA-A02 restricted peptides, and they also used one of the immunodominant HLA-B27 peptides, which were NS5B and 2841. So they use these peptides to stimulate the expanded CD8 T cells. So they use those peptides to stimulate the CD8 T cells, and in Figure 1A is the representative FACS plot. And hopefully you can see that as you increase the concentration of peptides, there are more interferon gamma being produced by the CD8 T cells, as you can see in the top right hand corner of each FACS plot. So as you increase the concentration, you get more interferon gamma being produced by the peptide-specific CD8 T cells, and which you could see the quantification in B and C. So in B, it shows the response curve. So you kind of plot the concentration of the peptides on the x-axis and the percentage of CD8 T cells, of the maximum of the CD8 T cells that produce interferon gamma here. And you could see that as you increase concentration, you get gradually you get more interferon gamma positive CD8 T cells, and hopefully you could see that for NS5B2841, which is immunodominant epitope. Surprisingly, you need a higher concentration of peptides in order to get the 50% of the maximum response compared to the less immunodominant HLA-A02 peptide, as you could see here. And in C, you can see the quantification. So clearly, at least in the chronic individuals, the functional avidity as measured by the interferon gamma production or interferon gamma positive CD8 T cells was a lot lower than the HLA-A02. So HLA-B27 restricted CD8 T cells had a lower functional avidity than the HLA-A02 restricted CD8 T cells. They also tested, so they also performed the same experiment on CD8 T cells from patients who cleared the virus spontaneously, and here they found that there was no significant difference between the CD8 T cells restricted to either of the HLA alleles. So what this figure showed was that the protection offered by the immunodominant epitope was not due to the superior functional avidity, and it's probably due to other factors. Yep. So in this figure, in Figure 2, they wanted to examine the multifunctionality, or the number of cytokines that CD8 T cells can produce by the CD8 T cells restricted by HLA-A02 and HLA-B27 alleles. So in A, upon the peptide stimulation, you could see that that the CD8 T cells that were restricted to the either alleles produced the same array of the cytokines. They produce quite a low amount of interleukin 2, but for CD107, which is LAMP1, which is indicative of the cytotoxicity, and interferon gamma as well, they produce similar concentration of interferon gamma and MIP1 beta and TNF alpha. And in B, which shows the multifunctionality, so the number indicates the number of cytokines being produced. So here you have, population of CD8 T cells that were producing 1 cytokine, 2 cytokines, 3, 4, 5 cytokines, and so on. And you can see that the two donut plots were very similar, a similar proportion of cells producing 3 and 4 cytokines and even 2 cytokines. So there was not that much difference, and in C you could see that there's not much difference at all. So they all produce similar concentration of cytokines, apart from this population of CD8 T cells. So this population of CD8 T cells were producing these 3 cytokines simultaneously. Well, CD107A is not a cytokine, but so they were producing CD107A, interferon gamma, and MIP1 beta simultaneously. So for this, so in this case, the CD8 T cells that were restricted to HLA-B27 were producing more of these cytokines. So in this figure, they wanted to find out whether the HLA-B27 restricted CD8 T cells had superior antiviral efficacy than the HLA-A02. So what they did was they incubated the HLA-restricted CD8 T cells with a cell line that is expressing either the HLA-A02 or HLA-B27 alleles, and then they measured for the ability of the CD8 T cells to inhibit the virus replication using the luciferase unit as a readout. And hopefully you could see that as you increase the concentration of the peptide, which you increase the number of CD8 T cells producing interferon gamma, you could see that there's a gradual decrease in the virus replication. So you see no virus replication at this concentration for HLA-A02, but for HLA-B27 you could still see some virus replication at this concentration. So basically what it is showing is that at least in the chronic individuals, the HLA-B27 had an inferior antiviral efficacy than the HLA-A02 restricted CD8 T cells. So they then so they did the same experiment on the cells from the resolved patients, and here what they found was that it's actually the opposite. So what they found was that the HLA-B27 restricted CD8 T cells had a slightly more antiviral efficacy than the HLA-A02 restricted CD8 T cells, but it was not that much more. So if you were to compare, this with this, it was not that much more superior. So they kind of dismissed it, and they're basically saying that the protection conferred by the HLA-B27 epitope was not due to the higher antiviral efficacy of the CD8 T cells, and it was due to other factors that they set to find out in this study. So in this figure, they wanted to find out whether the superior immunodominance of the HLA-B27 restricted peptides, the NS5B, was due to them having more naive precursors to the HLA and peptide complex. So in A, so what they did was they showed that in A, so they enriched for the peptide HLA restricted CD8 T cells. So they used tetramer. So they enrich it and stain for the naive precursor with different markers. And then they analyzed by flow cytometry. And what they found was that there was no difference in the number of CD8 precursors that were specific to these HLA and peptide complex. So the previous studies in HIV have shown that the antigen processing is important in the CD8 T cell response against the HIV. So what they did was they wanted to find out whether this was also the case with Hep C. So they measured the rate. So they measured how these three peptides, so the two A2 restricted peptides and the one B27 restricted peptides, were being processed by both forms of proteasomes. And hopefully you could see that by the lack of the black bars, which indicates the epitope containing fragments, you could see that the B27 restricted peptide was being very efficiently processed by both forms of immunoproteasome. And here in C, you could see that there was no epitope containing fragments here at all. So there was no ARMIL. So if you're looking for ARMIL, you could not see them at all. So because so what they did was they reduced the concentration of immunoproteasomes by fourfold. So they had fourfold less concentration. Sorry. They had fourfold less concentration of the proteasomes, and now they could see the epitope containing fragments being produced as shown by these symbols here. So basically what this graph is showing is that the HLA-B27 restricted epitope, the immunodominant epitope, was being efficiently digested by the proteasome. So they hypothesized that the flanking regions of the HLA-B27 might play a role in the antigen processing. So what they did was they swapped the two flanking regions of the B27, the immunodominant B27 flanking regions, with the flanking regions of the less dominant flanking regions from the A2 NS5B. So basically they swapped these two guys here with these two guys here, and as you could, and they named it B27 in A2. So basically this is the immunodominant epitope, and if you look at the flanking region, you can see that the flanking regions here are identical to the flanking regions here. And you have the A2 and A2, which is the original peptide. Then you have the original peptide here. I think the difference between this and this is the concentration of the proteasome. And as you can see that, so if we just look at first look at the B27 in B27, so the original B27 peptide at a high concentration of proteasome, you could see that this peptide is being processed very efficiently by the proteasomes, as you could not see the undigested peptides at all. And compared to the B27 in A2, so once you remove the flanking regions and replace it with the flanking regions of the less immunodominant epitope, you kind of reduce the efficiency of the antigen processing. The original B27 epitope was being digested very efficiently, but as you increase the concentration of proteasome, you see a more rapid processing. So next, they wanted to see whether the peptide fragments produced by the proteasome can induce the expansion and stimulate the CD8 T cells. So here they incubated the CD8 T cells with the peptides. So with 7 peptides actually, but in A is the representative facts plot showing incubation with 3 of the peptides, and hopefully you could see that as you increase the concentration of the peptides, you see more CD8 T cells producing interferon gamma, as shown in the right quadrant here. So that's the representative facts plot. And in B, so they use each of these fragments to stimulate the CD8 T cells, and what they found was that with the non-specific expansion, so these were CD8 T cells that were expanded using the antibodies and interleukin-2, and you could see that a lot of these fragments can induce the activation and expansion of CD8 T cells. So this was not as sensitive because it was non-specific, and because epitope-specific CD8 T cells only present in a very small number at any given time. So if you just look at the y-axis, so what they did was they specifically stimulate the CD8 T cells, meaning they expanded the CD8 T cells with all of these peptides, and then they individually test for each of these peptide ability to stimulate CD8 T cells. And what they found was that each of this peptide can indeed stimulate the CD8 T cell expansion. So the previous results have shown that the HLA-B 27 restricted epitope, the NS5B2841, was being efficiently processed by the proteasome. So what they then wanted to find out was whether it translated into more peptide fragments being presented on the surface of antigen presenting cells, and they use the activation of CD8 T cells as a readout. So what they did was they infected the antigen presenting cells with the vaccinia virus construct containing the Hep C polyprotein. So they infected the cells and incubated for 0 to 4 all the way to 24 hours. Then they incubated for 5 hours with CD8 T cells and measured the interferon gamma. So cells that were specific to the epitope will produce interferon gamma, and hopefully you could see that the longer you incubate the CD8 T cells with antigen presenting cells, the easier the interferon gamma can be identified. And you could see that on the in figure 8c as well. So if you were to plot it over time, the response over time, hopefully you could see that the HLA-B27 was efficiently being processed, more so than the HLA-A02, because it took the peptide less time to reach the cell surface and stimulate the CD8 T cells compared to the HLA-A02 restricted peptide, which needed approximately 4 more hours to stimulate the same number of or percentage of CD8 T cells. And hopefully you can see that in D as well, so you can plot the results in a chart, and you could see that at 2 and 4 hours there are more interferon gamma being detected when cells were infected with HLA-B27, and in 8 hours, after 8 hours, things became equal, because it had the HLA-A02 had enough time to catch up. And in E, they did some mathematical equation which translates into the amount of protein being presented on the cell surface of the antigen presenting cells. And here what you could see that at 2 hours and at 4 hours there were more fragments containing the HLA-B27 epitope being presented on the cell surface compared to the HLA-A02 peptide. But at 8 hours, things became equal again. So next I wanted to find out whether this was due to the proteasome. It was more like a proof of principle. And so what they did was they inhibit the proteasome, so that would inhibit the processing and antigen presentation, inhibit or slow down even. And you could see, so when cells were added with the epoxamycin, which is the proteasome inhibitor, you could see a significant reduction in the activation of CD8 T cells, which means it was a significant reduction in the antigen processing, as you can see the difference between the two. So it was proteasome dependent. So to summarize, and this is according to my understanding, and I've drawn these figures. So if I was to summarize this in one sentence, it would be the HLA-B27 restricted epitopes are processed much more efficiently by the proteasome. So if you can just go through that. So here on the left we have the HLA-A02 restricted epitopes. So we have the antigen presenting cells, and then which have been infected with the Hep C virus, which produces HCV proteins, and the proteins are chopped up or digested by the proteasome as well as immunoproteasome as part of the housekeeping, as well as the immune response. So the proteins get chopped up into fragments, and some of these fragments, which are specific to the protein encoded by HLA-AL2, so the fragments get presented on the cell surface, which can then activate antigen specific CD8 T cells. But in the case of HLA-B27, this process is much more efficient. So within 2 to 4 hours, you get more of the HLA-B27 restricted epitopes being made and presented on the cell surface of the antigen presenting cells, which can then activate CD8 T cells. So basically the the processing of HLA-B27 restricted epitopes is much more efficient and has a higher kinetics than the HLA-AL2 restricted epitopes. Yep, so here we come to the final part of the journal club, and I would like to name this the figure of the paper. And if there's a figure that you would like to memorize out of the whole paper, what you would choose. Personally, I would choose this figure, the figure 6, which kind of shows the efficiency of the processing of the B27 peptide. So this line here represent the B27 epitope, the B27 peptide at a high concentration of proteasome. So you could see that it was being efficiently degraded, and that's the same with the at a low concentration of proteasome. So these two peptides are the same, but just had a different concentration of proteasome. But if you were to compare that to the A2 restricted epitope and the B27 in A2, which was the B27 epitope, but with the flanking regions of the A2 restricted epitope, you could see the difference. So you could see the difference in these two here and these two here. So this is my figure of the paper. Thank you.
Hi guys, so thank you for going through this paper with me. I hope you enjoyed it. I did anyway. And yeah, so do you have a question regarding this paper and the data that were shown in this paper? If you have a question, just leave me a message in the comment section. And do you have a paper that you want me to go through? Please be immunology related. So yeah, please don't forget to press like and subscribe to my channel. And yeah, see you next time.